A very good afternoon, Manager. A warm welcome to all the dignitaries, esteemed guests, dear participants, and colleagues. Today, we have gathered here for the validatory function of World Bank ICAR funded NHP CAS training program on genomics of agriculturally important pathogens, microbes, and insects, which has held from 22nd July 2024 to 2nd August 2024. So in this occasion, I invite Dr. Antikumar sir, the course director, to welcome Dr. T.R. Sharma sir, DDG Crop Sciences ICR and Director of ICR IRI. Now may I call upon Dr. Subramanian sir to welcome Dr. C. Vishwanathan sir, Joint Director of Research and Coordinator of NHP CAS program. I would like to invite Dr. B. Ramakrishnan sir to welcome Dr. M. Saharan sir, Head Division of Plant Pathology. To Saharan sir, please. I also request Dr. Ramakrishnan sir to welcome Dr. M. K. Thilan sir, Head Division of Entomology. Thank you, sir. To proceed with the program, I would like to invite Dr. M. Saharan sir, Head Division of Plant Pathology, for the welcome address.
Thank you, Dr. Rajna. Good afternoon to all. Respected Chief Guest of today's function, Dr. Tilak Raj Sharma, DDG Crop Science and Director IRI, Dr. C. Vishwanathan, Joint Director Research IRI, Dr. D. Low, Head Entomology, Dr. Kumar, Principal Scientist Plant Pathology, Dr. Subramaniam, Dr. Deba Kamil, Dr. Ranjana, and dear participants. Today we feel very lucky that our Honorable DDG Crop Science and Director IRI, Dr. Tilak Raj Sharma, has accepted our request and is with us for this valedictory function. Sir, we welcome you from the core of our heart. I personally, as well as from the organizers from all the three divisions, entomology, pathology, microbiology, and on behalf of all the participants. So we welcome you, sir, from the core of the heart. A warm welcome to you for accepting our invitation and to come here to grace this occasion. And for the participants' benefit, you all know already about Dr. T.R. Sharma, a eminent scientist. I have the opportunity to meet sir from last 25 years, and sir PhD gold medalist from Palampur. After that, postdoc from Alberta, Edmonton, Canada. Then sir has the very significant work on rice blast. So sir is basically plant pathologist and then molecular plant pathologist and very renowned biotechnologist, bioinformatics. So sir, when joined in 99 at NIPB as senior scientist, and after that established the bioinformatics lab in NIPB, and from 2014 was also the director of NIPB. After that the director NABI in Mohali, and sir was also the executive officer there, other institute, CIAB, Center of Innovative and Applied Bioprocessing. And during that period, these two autonomous institute of the DBT, so sir performed very well there, and we were having many news from the media and many meeting with the Prime Minister. So those institutes are also excelled very well under Sir leadership. Now from 2020 January, Sir is DDG Crop Science and also guiding IRI as DDG from last 4 years, and from last 1 month also Director IRI. So Sir, main area is plant disease resistance, genomics and bioinformatics. So Sir, thank you very much for coming and to be the chief guest for today's function. Your blessing certainly will guide the student to excel in their career in the research or teaching or in other fields. We will feel benefited from your presence, sir. Thank you very much, sir. We also welcome our worthy Joint Director, Dr. C. Viswanathan. And Dr. C. Viswanathan, a very renowned scientist in plant physiology and molecular biology. And sir, you know, during your interaction during last 10 days with the lectures and other, and sir is NAS fellow and other many recognition in the agriculture science, in physiology and molecular biology and genome editing. A multi-institutional project sir is leading, and this NIHAP under the leadership of the Dr. Viswanathan as PI, we have organized like 35 training program. And in this training also there are 36 students and 250 students are also online. So sir, thank you for accepting our invitation. This is your own program. We welcome you for this. I also welcome Dr. Dillo, Head Entomology, to be available and to provide the guidance for this training program. And welcome to all the participants who are very important. There were 500 participants, and out of that you have been selected. You are 36 from 18 different states. So very diverse group. And I was in the first day also. And today in valedictory, it looks that you are more confident today from your face. So all the best wishes to you. I welcome you all for this. And welcome to all other, the organizing committee member, Dr. Deba, Dr. Ranjana and others for this. With this, I thank you all for this. Welcome again each and everyone for this training program. Thank you very much.
Thank you very much, sir. And a very heartfelt welcome to you also, sir. And may I now invite Dr. Anandkumar, sir, for briefing about the training program held now.
Good evening to all of you, the most respected director, joint director, heads of the divisions on the dais and delegates of the dais, and my fellow organizers, Dr. Subramaniam, Dr. Deepa, Rachna, Suresh, Dr. Ramakrishnan. Sir, this is 35th program, sir, we are conducting. And these 35 programs, we started sometime in 2018, the very first program. It started with the genomics of agriculturally important plant pathogens and microbes. And almost the same title, we are ending this whole program. But we added the insects here, genomics of agriculturally important pathogen, microbes, and insects. So it's a big learning for all of us as a team, cost team. And we trained, I would say proudly, hundreds of students representing various state agricultural universities and general universities on the core areas of genomics. That was our confidence, and let us listen from the students what they really learned. So in a nutshell, the entire team of students, they represented around 19 states and 18 universities, and the response was so immense. We received around 455 applications, and we had a slot of only 25. And I convinced our JDR shop to increase the number from 25 to 36, and he generously agreed, albeit a lot of difficulties in accommodating them in the hostels. But eventually around 36 of them are here. They attended the program offline, meaning in the physical presence with us. Since the number was so high, we left with no choice but to launch an online parallel activities also. So we streamed the entire program, the 10 days program, in YouTube channel online, and the number of participants were around 230 plus. So all these programs, whatever we did from the day one, the inaugural day and today, it is being recorded and it is being webcasted. So people who are connected through online can witness the whole event. The highlights are, we had around 24 lectures in the forenoon of all 10 days. That started on Monday to Tuesday, Friday, typically the first week, the second week also, again from Monday to Friday. Today is Friday, the last day. 24 lectures. Most of the lectures were focused on one or other genomic aspects. It could be the genome sequencing strategies or transcriptomics or metabolomics. It covered the variety of omics topics. I'm sure the lectures, the students would have enjoyed all those lectures, gained a lot of information, the theoretical information, and that was all about the theory. And the afternoon session is very, very important from the students' perspective. It's all practicals. And we tried our best to inculcate the genomic knowledge, the genomic literacy, I would say, right from how to extract the genomic DNA up to how to analyze them. So the major highlight of this 35th program, I would say very proudly, we demonstrated the whole genome sequencing of a bacteria and also a virus using nanopore sequencing technology. I think we divided the whole group into 2 batches, and the 2 batches are further subdivided, and each student was given an opportunity to witness how the genome sequencing has to go on using the long read sequencing technology, that is nanopore. So we dedicated 1 full day for this genome sequencing practical. I think that day, if I am correct, it was on 29th of July. We started at 9 o'clock and it went up to 5:30. So that was a major highlight. For the audience also, this batch is really privileged because we wanted to do this in the previous training program because the facility was still coming up, so we couldn't do it. Since the facility was in place, we could do it to all of you. I'm sure, if not everybody, I'm sure the substantial number of students might have got the theoretical account of how to do this genome sequencing. And those who represented IRA can do this genome sequencing on your own. You are most welcome. So the 3 domains we covered in this training program, microbes, pathogens, and insects. I think the lectures were really very rewarding. Most of the lectures I happened to witness online. When I'm here, I used to listen to all those lectures. Another major highlight, sir, we roped in an industry. The industry, they are one of the oldest industry in the world, the Merck, and they also acquired top-notch molecular biology service providers in the world, right from Sigmas and other companies. They were here yesterday. I think all of you witnessed their lecture also. They really delivered the major genomic breakthrough in biological sciences. And they also exposed all the students to some of the cutting edge protein gel electrophoresis. So that was a highlight of yesterday. I really thank Merck people for joining hand with us to teach our students. In addition to that, we also brought experts from outside, CPRA Shimla and University in Bhopal, and they also spoke in excellent lectures on species concepts and qPCRs. I think overall, let us listen from the students how much they really benefited. That is in a nutshell about the whole program, sir. So this is the last program, and as a team, we really wanted to make it one of the best. Let's listen from the students how much they learned from this program. So once again, I thank all the students and all the support what we received from the administrators for making this program one of the, in my opinion, a successful program. With that, thanks for the organizers for providing me the opportunity to narrate about the training program. Thank you. Yeah.
Thank you, sir, for a great elaboration. And now let us hear from the students. And we will be rating ourselves from you. So I request the students, participants to give the feedback over here. And I invite you all. I mean, some of them were ready. Yeah, it's open to everybody. Yes, sir.
Yeah. Worthy Director, sir, and all the organizing NAHP cast members. Good afternoon, everybody. Myself, Falak Fayaz. I'm a PhD student from the Division of Plant Pathology. So today, I would be reflecting back upon the experiences that we witnessed from past 12 days. I would like to call myself a freestyle writer. Okay, I write upon everything and anything, or we can say vice versa. So today I have prepared this excerpt on the experiences that we witnessed from the past 12 days. So it goes like this. When I came, I was a novice to the practices, processes, and to the code of genomics. But having said that, each exposure helped me to think distinct, odd, and unexpected. 12 days, 18 states, and a bunch of scholars gathered within the walls of Discovery Center, just seeping in the theorem and trying to find a why of their life. While delving deep into the strings of DNA, I met the SNPs and greeted the variants there at the base level that got to know there exists a world beyond the phenotype just at the brim of exploration. From base calls to contigs, I got assembled and faded into the virgin who is ready to leave the exui and gain the leap. Dancing on the waves of genomics with CRISPR as our harmonic mean, every session widened the horizons and each exposure opened the windows to the novel questions and path-breaking finds. From Petri dish to the mesmerizing world of in silico, I am in complete awe for the sapient intellect and in bow for the scientific commune. With this, I would like to extend my heartfelt gratitude to this scientific society who rendered this hands-on service for we people, and I am totally in debt to the NAHP cast coordinators for organizing and selecting us for this coveted, wonderful and prestigious experience. Thank you so much.
Please come forward. No. Falak, it was excellent. You are really a freelance writer.
Good evening, everyone. I'm Rajni Kumari. I'm from MPUAT, Rajasthan College of Agriculture, and I'm doing PhD here. And first of all, I would like to thank you for choosing me. And as we gather here today on the last day as our training, I want to take a moment to express my deepest gratitude to all the members who organized this incredible training program. And all members who are directly or indirectly involved in this training program. Your hard work and dedication truly made this experience unforgettable. Over the past days, each and every one of you has taught us invaluable lesson. You have shared your knowledge, expertise, and passion, making sure that we leave this training with a wealth of information and skill. Your commitment to our growth and development has not gone unnoticed, and we are immensely grateful for that. Thank you so much to all for such a fantastic training experience. We have learned a lot, and we appreciate everything you have did for me. Thank you so much. Have a nice day.
Okay. Thank you very much. Because of the time constraint, means if somebody has to sell something which just you can sell 1 or 2 sentences, otherwise we'll move on forward. Okay. Otherwise you can come to us and you can personally also tell to modify the programs because of the time constraint. Okay. So may I call upon Dr. C. Vishwanathan, sir, Joint Director of Research and the person behind all this NHP gas training programs for the address.
Respected Dr. T.R. Sharma sir, the Deputy Director General Crop Science ICAR, and also the Director IRI. And I am sure we could not have find any other better person for giving you the certificate on the topic which is so close to Dr. T.R. Sharma sir's heart, and also he has made immense contribution. Everyone might have heard about RISE genome sequencing project. Sir has contributed immensely to the RISE genome, wheat genome, and different genome projects that has been published in the Nature and Science. So he is well-known genomics scientist. So you are all blessed to have a certificate from sir today. Sir, we are very thankful, like despite there are a lot of engagement, sharing two big responsibilities not small, like as a DDG and as a director IRI, you know, every minute is so crucial. But despite that, he gave time for all of you just to meet you and greet you and give you a certificate. So this we are very thankful to you, sir. And so I don't want to give any speech here. Already I have given one lecture on genome editing to you in the inaugural program. Also I talked to you. So as I told, phenome is interaction of G into E. That's what we study. So in the E, several components are there. So all this you have studied the microbes and insects. So everything comes in the E component. Earlier we thought only E is only the plant is providing the G component. So now the plant genome interact with all this genome, and that ultimately decides the phenotype. To understand this, the genomics is very important to understand how the host and pathogen interact, and how we can manage the pathogens, or how we can effectively use the beneficial organism for enhancing the productivity while maintaining the environmental sustainability. The goal of this project was to enhance the skill of the student to harness the global gene pool. So now it is the entire gene pool we can transfer from one organism to another organism, and better understanding of the genes is very critical. Genome to genes, that is very critical for utilizing this global gene pool effectively for the benefit of the human kind. So that's why this program is so important. Only request to all of you is that you have come here and you have got the benefit of attending this training. Go and spread the knowledge. In your department, maybe you can give a 1 hour seminar. Like if we go to foreign countries, when we return, we give 1 hour seminar. So in the same way, you can also give some seminar, and you can share your the training manual and other resource material with your fellow students, so that they will also get benefit. And also you can tell about the YouTube channel where we have recorded all the lectures and broadcasted. So I am sure that will be very useful. And I thank only 2 students, they have got opportunity to speak, and other students also, they have not complained despite all the limitations that you have in your accommodation and everything. You those feedback you will give, we will try to improve in the coming times. But I am sure you have got a lot of intellectual input, and that help to save your career. And you maintain this WhatsApp group, so that every time when you have any doubt, you can put a message. So either the faculty will respond, or one of your fellow colleague will respond. So continue this interaction in the coming future. If you have any questions related to genomics, you are free to contact any of the faculty member, and we will try to address all those questions. So with this, I thank the organizer for making the excellent program. Sir, the limitation of number is mainly due to the accommodation, sir. So hope with your help, maybe in the next EFC we will construct one big trainees hostel, sir. Then if it is there, then we can accommodate more students for training. Thank you so much, sir. Thank you.
Thank you very much, sir. Sir, now it is the time for certificate distribution for the participants, sir. May I request the dignitaries on the dais to come forward for the certificate distribution. So I'll call your names and then accordingly please come. Amar Singh. Manish Yadekar. Tinta Konyak. Veeramanikanda Reddy. Saravanan S. Rod Shubham. Satendra Kumar Sharma. This is Rod Shubham. Satendra Kumar Sharma. Shubham Kaundal. Shubham Kaundal. Himanshu Jain. Tirumala Geetika. Sayanthini Nath. This is Sayanthini Nath. Vaishnavi. Yashaswini Goswami. Kavya M. Hairmat. Sakshi Sharma. Vandana Hijam. Jonnada Likita. Miss Kanuri Komala Shiva Katyani. Rajini Kumari. Rajini Kumari. Deepika Bansal. Arushi Aran. Jinisha Blessy. Pratnya Vasant Uke. Rajshri Karanwal. Rajshri Karanwal. Prajal Gowda. Lognathan. Prajal Gowda. Lognathan R. Viresha P. Coach Hania. Ashwini JH. Ashwini JH. Swagata Sarkar. Aditi Singh. Ilangovan M. Ruth Olivia. Isha Pokare, Isha Pokare, Aravind Kumar, Falak Fayaz. Sir, we also have our course associate who has worked these 14 days hard. So we just want to appreciate them also, sir. Please request from our side. Mr. Pavan J. Mr. Rudra Gowda. Chavar Rajarushi. Mohammad Javed. Vinod Chauhan. Vel Murugan. Prashant Patidar. Prashant. Thank you very much all. It's our privilege to have Dr. T.R. Sharma sir with us. Sir, we are requesting you for the chief guest address and valedictory address together.
Good afternoon to all. Dr. C. Viswanathan, Joint Director Research, ICAR, New Delhi. Dr. M. K. Dhillon, Head of Division of Entomology. Dr. M. S. Saharan, Head Division of Plant Pathology. Dr. A. Kumar, Post Director. Dr. Diva Kamil, Dr. Subramaniam, dear students, boys and girls. I'm really happy to be here this afternoon to participate with you on the valedictory function of this very important course, which was conducted by 3 divisions together. It was really a very good initiative by all the head of the divisions of microbiology, plant pathology, and entomology to come together and try to train the students on various aspects of genome sequencing. This topic is really very dear to me because, as told by Dr. Viswanathan, not only I was in, and lambda was the only organism which sequencing was done only manually. And subsequently, when lambda DNA sequencing came into being in 1999 by a company applied by systems, then was the automated Sanger's dideoxy method of sequencing and using computational and machine sequencers. Automated DNA, have you seen the sequencing came into being in 1999 should have shown by a company applied IPV. Still, I think line systems in some of the labs machine. The automated Sanger's dideoxy methods of sequencing and using that machine. Using computational and sequencing sequencers. Have you seen the sequencers? During those period, when you should have shown that sequencer, India also entered, still I think lying there in some of the labs. International Consortium of Sequencing Rice Genome. And using that machine, and you will be surprised to people know who are sequencing eukaryotic genome sequencing now whole genome sequencing. During those like period in 2000, then India also and whole genome is cloned in International Consortium of Sequencing Rice Genome or back vectors, bacterial and artificial. You will nice to know that the genome which you are sequencing whole genome sequencing in a fragment was not like say under 50 to 200 nucleotides. First of all, whole genome, and then all those is cloned from a library pack or YAP vectors, select back vectors, bacterial, artificial chromosome specific DNA or yeast artificial chromosome physical map in a fragment of say under 50 to 200 linkage map or genetic map, and then all those back clones from a library in terms of are selected organs with the help of physical chromosome specific DNA markers to construct a physical map. So, that's where we linkage map and rice map was where the map distance is measured in terms of 42,000 organ clones. Whole rice is a physical map where distance is measured in terms of base pairs. 30 labs across 10 countries. That's where we started. And rice was cloned in, and the whole genome was distributed. Whole rice distributed. 10 countries were involved. For example, India was across 10 countries, 250 scientists across the globe. Chromosome number 11 of rice. The whole genome was distributed. It's very interesting. Distributed Delhi University and South Campus. Chromosome wise. So we further divided that India was sequencing into two parts. Two parts campus. One was we further the IRI and another was with the Delhi University South Campus. So even for sequencing that small fragments, if you see from today's context, it took us 5 years, and we had more than 25 GRF, RA researchers. We all scientists were working. We were going abroad. I went to Cold Spring Harbor Lab two times, 2001 and 2004. Cold Spring Harbor Lab is Watson's lab, if you remember, where Hershey and Chase for the first time showed that nucleotides is a genetic material. And Barbara McClintock discovered jumping genes or transposable elements in maize in the same lab. And for DNA structure, double structure, the first presentation was made by Watson and Crick at Cold Spring Harbor conference. So they were human genome. So I was doing rice genome sequencing in human genome sequencing lab. And you will be surprised to know that picking up individual back clone from 96 well plates. Have you seen 96 or 384 well plate? Okay. So that plates we used to fill with robotics there in US. Here we were filling manually with multi-channel pipettes. And at the end of experiments, if you see in the evening, our thumbs and fingers were swelling because of a lot of pipetting and processing samples. And there you don't get assistance. You have to do everything at your own lab. There I learned how high throughput sequencing is done. So till 2005, this Sanger's dideoxy method of sequencing was very common, applied by a systems machine. But after 2005, there was a complete transformation in developing sequencing technologies, and next generation sequencing technologies appeared in the scientific scene. Okay, one was Illumina, another is PacBio, another is now Nanopore came only in somewhere in 2015 or 16. So the whole rice genome was sequenced by 250 people in 30 labs of 10 countries across the globes in 10 years time. And the size was 420 million base pair. But if you see the Nature paper in which that was published, genome was published, we could sequence 389 million base pairs. Because you should also know that rice has 12 chromosomes and all centromeric regions and telomeric regions were not sequenced. Now, as a biologist, you can ask question why those were not sequenced. Because of heterochromatic nature of those regions, highly repetitive. And it's very difficult to sequence repetitive genome. Now, if you see the basic chemistry of sequencing. For sequencing, DNA has to decoil first, isn't it? It's a double stranded DNA. So you make it single stranded form. Primer is attached to one of the fragments, and then it start coding from reading the template. So that heterochromatic region is unable to decoil because of why? It's a GC rich. Now you have to remember the DNA structure. AT is attached with 2 hydrogen bonds, whereas GC with 3 hydrogen bonds. So 3 hydrogen bonds are more robust than 2. So it's all chemistry. But with all those difficulties, the rice genome was sequenced, and as told by Dr. Vishwanathan, paper published in Nature, and you see that paper, Google search, it's freely available. It's a gold standard of any plant genome after Arabidopsis. Only 6 pages paper, and a lot of supplementary material. Then after 2006, next generation sequence came into being, and then we started sequencing tomato genome. India as a country, along with again around 15 countries of the world, and it was also a large network. And luckily, tomato also has 12 chromosomes. The genome size is around 740 million base pairs. So there we sequence full chromosome. So because we became expert of sequencing rice genome, so India took whole chromosome 5. And using hybrid technology, clone by clone approach, as well as next generation sequencing technology, and both hybrid technology was used. And in that paper, which was again appeared in Nature in 2012, it's 8 pages paper with 175 175 supplementary pages. So now you can imagine how much data was generated there. Then we took up third genome, pigeon pea genome. The genome size is around 480 million base pair. And there we used only next generation sequencing technology, and the whole genome was sequenced by Indian worker, only IRI or NIPV. And there we used only next generation sequencing technologies, no clone by clone approach, no hybrid method. So why I'm telling all those things? Because how systematically this advancement in technology of sequencing progressed from 1972, when sequencing was developed by Sangers and Maxim and Gilbert, and then 99 automated, and then next generation, and then nanopore 2015 onwards. Using nanopores, we sequenced a wheat variety C306 when I was at Mohali, and around 10 land races of rice from Northeast. Kola Joha, who are from Northeast, they might be Kola Joha or Manipuri black rice, that we sequenced, and that paper is in Genomics. So, and there we used nanopore sequencing. So starting from a gel to nanopore, which you have used, we used all the technologies for sequencing, but the most important information which you can draw from sequencing is to analyze the genome sequence available in your lab or in public domain. For that, computational biology or bioinformatics is very, very important. So the second training which I got in 2004 was on genome finishing and bioinformatics. Finishing means filling all the gaps from, say, short chromosome telomeric end to long chromosome telomeric end. From one end to another end, if there are certain gaps that you have to fill by manually sequencing, and that's very, very tough. There are very few people in the world who does that type of work, and they are named as finishers. They finish genome. Okay, so and here we introduced a course on bioinformatics. IRI, if you are, any of you are old student of IRI, those who took classes with me in 2004 and 2006, 7, 8, we introduced a course on bioinformatics. That was very, very popular course. And in 2008, then I wrote a book on computational biology and genome analysis and bioinformatics, a practical approach. If you Google search, you will find that small book, which is based on my class notes, which I used to teach in the class. So that I converted into a book form and made it and asked these publishers to keep the price of book less than 200 rupees so that all students should buy it. He told me that I will publish a hardcover book with 1000 rupees price. I told no, you can't do that. The price should be less than 200 rupees so that most of the students anywhere if they are working. And I'm told that that book is highly sought and still it is available on Amazon. If you Google search and find with my name, the book is available, but I have not seen now for the past many years. But probably whenever I will get time, I will try to revise that because a lot of advancement has taken place in terms of genomics and computational biology. So you people are very, very lucky that you got to know through practical demonstrations how sequences are generated from a machine, starting from DNA isolation to preparing a sequencing reaction and then putting on a machine and then getting sequence and then analyzing sequence on computers. I don't know whether you have analyzed or not, and you might be told how to identify a gene within a sequence. Okay, if you use your theoretical, because you all are biologists, that's why I am talking all these terms. Had there been some other subject people sitting here, probably I would have not talked all these basic things here because you are students and you will also understand what I am talking here. So if you know the gene structure, based on gene structure, it is identified from the sequence because it has start codon, you know, it's a very distinguished 3 nucleotides, stop codon, then exonic region ORF, open reading frame, poly A tail at the end, and promoter region at the 3' end, isn't it? All these computational methods are based on all these 5 features of a gene, and based on these 5 features, the genome sequence is identified by doing comparative analysis in the database. So I'm sure that also has already been taught to you. If not, then you should know from the NCBI website. Many tools are available there. Tutorials are available on NCBI. And I learned many things from NCBI tutorials during my days when I was doing sequencing practically with my own hands. So you should also refer to that. Have you seen NCBI website? Have you seen PubMed? Yes, good. So there, if you go to NCBI, there are a very distinct sections on tutorials. And there you will learn so many, not only on genome sequence analysis, but also proteomics and 3D protein modeling. All those things you have to do. So that's what we did. Not only we stopped with sequencing, we cloned gene from rice. If you see, my whole work is from rice from 96 till date. I'm working on rice. That's true on one gene, PI54, which we cloned in 2005 from a rice line after our 10 years of work. So you need a lot of patience while working as a student and also as a scientist. Because we used what you call map-based cloning approach of identifying a gene. For map-based cloning approach, you only target those genes where phenotype of a gene is not known. You can see a gene only from its expression. Protein you don't know, the gene product you don't know. So you have to construct a linkage map and then reach up to the gene of interest and clone that, functionally validate that, and put in again susceptible lines, if it is a resistance gene, and then do all sorts of work which you wish to do. So my suggestions to all of you would be that after learning for 12 days, this type of technology you apply in your research. Nowadays, these are not very expensive science. Now, during our days, it was very expensive. Even to run a small sequencing reaction, it was costing thousands of rupees. Now, in thousands of rupees, you can sequence full genome. Okay, and bacterial genome, insect genome, or what else you did? Microbes, insect, and pathogens. All these three. Pathogen, I'm sure you have studied fungal genome. So the average genome size of bacteria is 4 to 5 million base pair. If you see in literature, the average genome size of fungal is 35 to 40 million base pairs. There are certain exceptions like Phytophthora, where genome is 240 million base pair. Rust, where genome size is 80 to 120 million base pair, depending upon which rust you are studying. And also insect genome size like Drosophila is 180 million base pair. So Drosophila is a standard genome which was sequenced for the first time in the year of 2000. It was the first genome of eukaryotic microorganism after Saccharomyces cerevisiae. Saccharomyces is a yeast. So after yeast sequence genomes, this Drosophila melanogaster was sequenced. And then subsequently rice, tomato, wheat, and now many genomes, and you do pan-genome analysis, you do multiple genome sequencing. So this technology is very, very important, and a very important technology now. Even I'm sure that you are also taught about transcriptome analysis and how do you derive transcriptomes, because after structural genomics, you have to do functional genome analysis by identifying individual gene and then looking at the function of each gene. Rice genome was sequenced in 2005, and after even 20 years of its sequencing, not more than 10% of its genes have been functionally validated. Rest of the 90% genes are still unvalidated. We don't know their function. So a lot of work has to be done by all young colleagues here, and I'm sure even your life career, there might be some other sequencing method or some approach which will come. And when you are standing here after say 30 years, okay, you will be sitting across the table, you will be narrating your story that you did one training at IRI division for 12 days and learned all those things, because these are the what you call basis of your progress in science. So whenever you get opportunity for any training related to your field, I will advise you attend those trainings. And also make contacts with your colleagues. Here you are 35 people from different around 19, 17, 18 states. So you make your good friends, exchange your ideas with them, and keep those contacts live for years to come. Okay, so my best wishes to all of you, and congratulations to the organizers for planning such a wonderful training. And I'm sure that in future you are going to use this training for the welfare of society and to the farmers. So best wishes to all of you. Thank you very much.
Thank you very much, sir, for enlightening the gathering with your experiences and insightfulness. And sir has traveled from very basics of genomics to applied parts in a very short time, sir, and we are all very fortunate to hear you, sir. Thank you very much, sir. Now it's time to pay our gratitude to our guest over here today. May I invite Head Division of Entomology, Dr. M.K. Dhillon, sir, to felicitate our dignitaries. Sir, I request you to felicitate Dr. T.R. Sharma, sir. Sir, again I request you to felicitate our JDR, sir. Thank you very much, everybody. And now we have come to an end to our 2 weeks long training program, and I invite Dr. Deepa Kamal ma'am, the course coordinator, to propose the formal vote of thanks.
So,
Thank you, Dr. Rachana. Good evening, everyone. As we come to close the enriching and enlightening training program on genomics of agriculture, important pathogen, microbes, and insect organized under the NHP CAS initiative by Division of Plant Pathology, Microbiology, Entomology at IRI, it's my honor and privilege to deliver the vote of thanks. First and foremost, I would like to express my deeply gratitude to our esteemed Chief Guest, Dr. T.R. Sharma, sir, DDG Crop Science and Director IRI, for gracing this occasion with your presence and your inspiring word that have added great value to this program, sir. As a special thanks go to our distinguished Dr. C. Vishnathan, sir, Joint Director Research and PIO of NHP CAST, for his constant support, encouragement, and guidance for this entire program. I would like to thank Head Plant Pathology, Entomology, Microbiology Division, Dr. M.S. Saran sir, Dr. M.K. Dillon sir, and Dr. Radha Prasanna ma'am for their all-time support and help for completion of this entire 10 days training. Also, the thanks goes to Dr. Renu Pandey ma'am, Head Division of Plant Physiology, for providing GS Heroic Hall and the logistic for entire training program. I would like to extend my sincere thanks to organizing committee, especially course director Dr. A. Kumar, Dr. S. Subramaniam sir, Dr. B. Ramakrishnan sir, co-director Dr. Suresh, Dr. Rajana, and training associate like Javed, Vinod, Velmurugan, Prashant, Rushi, Rudra, Shanu, and many more, for their constant instrumental for this arranging the training program, especially for the practicals. Thank you all of you. To my this participation, the PhD and PhD and PG student, your that ability to learn so fast and that knowledge and the skill you have acquired here to serve as a strong foundation for your future research and the professional endeavors. We are also grateful to NHP coordinator and the entire team for this continuous support, for providing us this platform to bring together as a diverse and talented group individuals. I also want to thank the team associated with online broadcasting for their hard work for conduct this training at the hybrid mode. I also want to thank the entire technical staff, administrative staff to run that training in the whole logistic, and you reflect and deeply appreciate. In the last, I want to thank all the person who is directly and indirectly associated with this training. Thank you so much.
Now I invite everybody for a group photo outside, and after that we will have the tea. But we'll go for the group photo outside this hall.